Thank you all for joining our session today titled Modeling Neural Activity in a Dish, Advancing Our Understanding of Nervous System Disorders. My name is Stacy Cavadal, and I'm the product manager here at Axion BioSystems. We know that all of you are great stem cell biologists, and you're making more and more intricate disease models. It's important to verify that those models are functionally active and understand how they impact neural signaling. Today I will give a brief introduction to Maestro MEA technology, and then you will hear from two speakers using stem cells and Maestro MEA to study mechanisms behind epilepsy and neuropathy. Understanding how a cell functions is critical to appreciating its role in the body and disease. The ability to transmit electrical signals is a key function of many cells. An MEA is a grid of small electrodes. When embedded in the surface of a well and using only basic cell culture techniques, researchers plate electrically active cells on top of the electrodes and measure their activity without disrupting the cells. MEA electrodes are sensitive enough to detect the activity of individual neurons. The signal shown here represents the voltage over time from a single electrode. Each time the neuron above fires an action potential, a spike appears in the signal. Spikes are simultaneously detected from multiple electrodes across the array, making it ideal to monitor activity throughout the culture and investigate network behavior. A simple way to visualize this network behavior is with a raster plot. Each tick mark represents the time, on the x-axis, that a spike occurred. Each row of tick marks represents the spikes from a single electrode. A well-wide raster plot shows the activity from all of the electrodes in the well, providing important insights into the behavior of the culture. By looking at different metrics that describe the firing patterns, such as activity, synchrony, and network oscillations, an activity profile of the culture is made. These activity profiles act as a fingerprint for the culture, changing based on the composition of cells in culture, their health, maturity, genetic makeup, or chemical effects. With the Maestro multiwell MEA, neural activity can be monitored from multiple cultures in up to 96 wells at the same time. Because the measurement is non-invasive, the activity can be measured from the same plates many times over months. With MEA, any cell culture lab can easily conduct important electrophysiological experiments, whether characterizing an iPSC-derived culture, studying development, disease progression, or screening compounds for safety and efficacy. The detailed functional data from MEA provides critical information. If you have any questions for our speakers during this session, please submit them during the presentation, and we will answer them during the Q&A at the end of the session. Our first speaker today is Dr. Evangelos Kiskinis. Dr. Kiskinis is an assistant professor of neurology at Northwestern University Feinberg School of Medicine. He also serves as the scientific director of the Stem Cell Core Facility and is the co-director of the Stem Cell and Regenerative Biology Initiative. Today he will present his work titled Epilepsy in a Dish, a case study in pediatric KCNQ2-associated epileptic encephalopathy.
Thank you for that kind introduction and invitation. I'm really happy to be here and talk to you about our efforts to establish models of pediatric epilepsy using induced pluripotent stem cells with the goal of understanding disease mechanisms and hopefully contributing towards the development of effective therapeutics for these devastating syndromes. So in my lab, we're focused on establishing human-based models of neurological and neurodegenerative disease. We typically recruit a patient of interest through one of our neurology clinics. We isolate some sort of somatic cell type from that individual, and we generate patient-specific induced pluripotent stem cells. We utilize CRISPR-Cas9 gene editing to correct disease-associated mutations and generate isogenic control iPSC lines. Once we have these pairs of lines in our lab, we in vitro differentiate them into disease cell types, including cortical GABAergic neuronal subtypes and cortical glutamatergic excitatory neurons. Once we have these cells in the lab, we use them to perform all kinds of genomic, functional, and biochemical analysis to identify disease mechanisms and hopefully patient-specific drugs. Pediatric epilepsy is a major public health concern. Each year, between 1 to 5 in every 1,000 children born are affected by some sort of seizure. In the most severe cases, babies start seizing within hours or days after they're born. The seizures, which result from aberrant electrical activity in the brain, are multiform, relentless, and sometimes they're exceptionally hard to treat. As a result, these children suffer serious comorbidities including cognitive, behavioral, and neurological deficits. Clinicians have no effective way of selecting the right anti-epileptic drug for each individual, and often resort to a trial and error approach which can have detrimental effects for babies that are in developmentally very sensitive stages. Pediatric epilepsy has extensive genetic diversity, with 50% of cases caused by de novo mutations in ion channel genes, which of course control neuron excitability. These include sodium channels and potassium channels, and today I'll focus on a very particular subtype of pediatric epilepsy associated with mutations in the potassium channel gene called KCNQ2. The KCNQ2 gene encodes a 6-transmembrane domain protein that interacts with KCNQ3 to form a functional potassium ion channel that is responsible for neuronal M current. There are currently close to 300 distinct variants associated with KCNQ2 epilepsy, and the problem is that we don't entirely understand the functional ramifications of these mutations on the ion channel itself or on the behavior of neurons. In fact, according to ClinVar, which is a public archive that reports on the relationships between human variations and phenotypes, there are a number of different classifications of variants. Some variants are cast as pathogenic, others are likely pathogenic, others are variants of unknown significance, some as conflicting, and several are as benign or likely benign. To address this variant classification problem, we've been collaborating with Al George and his group, who have developed a high-throughput functional assay based on the use of an automated whole-cell patch instrumentation. This approach relies on the heterologous expression of either the wild-type or the mutant KCNQ2 protein in a cell line that stably expresses KCNQ3, which is an essential component of the functional tetrameric channel. This is an example of the primary data, which represents the average whole cell M current recorded from hundreds of cells that express either the wild type or the mutant protein. The system allows us to collect measurements from hundreds of cells and screen through a high number of variants in a very short amount of time. We use the functional data to classify variants based on their pathogenic effect on the channel. This graph represents the relative percent shift in current density relative to the wild-type case, with a decrease marked in red on the left and an increase marked in green on the right. This representative data set showcases the effects of 41 KCNQ2 genetic variants. You can see that the overwhelming majority caused a reduction in current density, with only 2 causing a significant increase. Critically, variants associated with benign epilepsy cases, marked in blue, do not seem to strongly segregate away from variants associated with severe epileptic encephalopathy cases, marked in red. One potential interpretation of this analysis is that the primary effect of the mutation on current density is not a major determinant of clinical severity. A critical question that we can only address with iPSC technology is what are the functional ramifications of these channels on patient neurons, while we can rely on the neurons themselves to assemble the channel. To address this, we've been using the classification of from the heterologous expression model that I described to you before to select representative variants and analyze their effects in iPSC patient-derived neurons. In collaboration with John Millichap and other clinicians at Lowry Children's Hospital in Chicago, we have set up an IRB-approved protocol that has allowed us to enroll 20 children with KCNQ2-associated epilepsy. We've made induced pluripotent stem cell lines from all of these cases and have successfully corrected their mutation by CRISPR-Cas9 in approximately half of them so far. For the next few slides, I will present to you data from one such patient as a case study. This was a female individual with no known family history who was clinically diagnosed with epileptic seizures within 1 month after she was born. At the time at which she was recruited in our program, she was already 13 years old and she had significant neurodevelopmental delay. Clinical genetic panel testing revealed a single de novo heterozygous variant in the KCNQ2 gene, which resulted in an R to581Q amino acid change. At the bottom left, I'm showing you the sequencing chromatographs depicting the heterozygous mutation in the middle, in the case of the patient, and one of the isogenic control-corrected clones that we used in our study. At the bottom right, I'm showing you some basic characterization of the differentiated NGN2-based cortical excitatory neurons, all of which are glutamatergic, and all of which are earmarked with GFP so we can identify them in culture. One of the platforms we used to interrogate the excitatory neurons derived from this patient was the multi-electrode array, which allows us to record spontaneous activity from populations of neurons across a period of time in vitro, during which the neurons are continuously maturing and developing a functional network. This is some of the primary data. Critically, I'm showing you here in the top left that we compared a similar number of plated patient and isogenic control neurons. One of the first observations that we made was that the patient neurons exhibited an earlier initiation of spontaneous activity, shown here at the bottom with these raster plots for days 12, 13, and 15, and quantified up here as the number of active electrodes in patient neurons shown in red and isogenic control neurons shown in blue, as well as the mean firing rate for each one of the different genotypes. Most importantly, when we evaluated the activity of these neurons across several weeks in culture, we observed a robust shift in their firing behavior in the case of the patient cells. We specifically found that the mutant neurons exhibited a high number of bursts, an increased number of spikes per burst, as well as an increased percentage of all spikes that occur within bursts. This is shown here in the raster plots on the top left, as well as this in these quantitative measurements across time at the bottom. Collectively, these metrics are telling us that the patient neurons exhibit significantly enhanced spontaneous burst firing. We are excited about these findings because children diagnosed with KCNQ2-associated epilepsy exhibit a characteristic pattern in their brain EEGs, known as a burst suppression activity pattern. They specifically show highly synchronized firing, i.e. bursting, followed by intermittent periods of very little activity. In fact, when we represented the iPSC-derived neuronal data in a raster plot within the same 20-second interval as the EEG pattern shown here, we highlighted a burst suppression firing pattern which was highly reminiscent of the one seen in the patient EEG, essentially recapitulating epilepsy in a dish. This provides us with one phenotypic platform that can be used to interrogate disease mechanism as well as to assess therapeutic options. To better understand what is the mechanism that drives this shift in firing behavior, we use single-cell patch clamp analysis. In fact, when we analyzed on average 50 to 60 neurons per genotype across independent differentiation cohorts, we found that the patient cells exhibited an enhancement of action potential repolarization with faster action potential half-widths and enhanced fast after hyperpolarization potentials. Most strikingly, the post-burst AHPs were larger in patient neurons throughout the timeline of our recorded experiments. Using transcriptional and pharmacological approaches, we identified a set of dyshomeostatic changes driven by the functional upregulation of potassium currents mediated by BK and SK channels. On the left is quantitative real-time PCR analysis showing the upregulation of several potassium channel and other associated genes in the patient neurons after weeks 3 and week 5 in culture. On the right, I'm showing you that in fact, treatment of patient neurons with the SK and BK channel inhibitors apamine and paxilin rescued the phasic bursting, while the same treatment had no effect on isogenic control neurons. We next wanted to ensure that these rather unexpected changes we identified were indeed driven by the primary defect of the mutation on mCARN itself. To test this idea, we chronically treated control neurons with a specific mCARN blocker known as XZ991 and assessed neuronal activity by single-cell patch clamp, multi-electrode arrays, as well as by quantitative real-time PCR. We found that chronic inhibition of mCARN in control neurons phenocopies the alterations seen in mutant KCNQ2 patient-specific neurons. Interestingly, the effects of chronic inhibition were in some cases more dramatic than the ones seen on patient neurons. Moreover, while the transcriptional changes were not 100% overlapping between the patient neurons and the chronically treated control neurons, the overall dyshomeostatic mechanisms were concordant with our initial hypothesis. Everything that I've shown you so far concerns one particular individual. Critically, we have to determine whether these phenotypic alterations, as well as this dyshomeostatic mechanism that we identified, are more broadly relevant across other KCNQ2 patients. Using the same approaches that I described earlier, we're finding that multiple patients harboring distinct mutations in the KCNQ2 gene share the same enhanced spontaneous phasic bursting phenotype as measured on a multi-electrode array. Specifically, I'm showing you here 2 additional cases next to the R581Q individual shown here on the far right. Critically, these 3 cases have dissimilar levels of loss of function effects on mCARN density as shown here based on the heterologous expression system I described earlier. This raises several intriguing questions related to the origin as well as to the mechanism of dyshomeostatic adaptation in each case, and we're currently focused on addressing these critical questions. In summary, I've shown you that KCNQ2 channelopathy is an important cause of monogenic epilepsy. Many KCNQ2 variants have unknown clinical significance. Automated patch clamp can categorize KCNQ2 variants. KCNQ2 iPSC-derived neurons exhibit spontaneous burst suppression firing that is reminiscent of the EEG pattern for KCNQ2 patients. Dyshomeostatic functional upregulation of BK and SK currents is driving the bursting that we discovered in patient neurons. And the question we're currently focused on is whether these KCNQ2 iPSC-based models can indeed be used to assess the efficacy of small molecules targeting potassium channels. Thank you for your attention and time. Thank you also to Dina Simkin, who's been spearheading this work in my lab, as well as to our invaluable collaborators, particularly Carlos Vanoy and Al George, as well as our funding agencies, the NIH, which has been supporting this work through a U54 Center Without Walls grant, as well as the New York Stem Cell Foundation that has been supporting my lab.
Thank you, Dr. Kiskinis, for that really interesting and informative presentation. Our next speaker is Dr. David Belair. David performed the work he's showing here while employed by Bristol Myers Squibb, formerly Celgene, in Summit, New Jersey. He is currently a senior scientist at AbbVie in North Chicago, Illinois. Today he will discuss his previous work titled Modeling Peripheral Neuropathy with Stem Cell-Derived Peripheral Neurons on Multi-Electrode Arrays.
Thank you for the introduction, and thank you to Axion for the invitation to present on our research. Here we used an approach to model peripheral neuropathy using stem cell-derived peripheral neurons. And one of the tools that we used to do that was multi-electrode array, the Axion Maestro system. Just a quick disclaimer, this work was performed while I was employed by Bristol Myers Squibb, formerly Celgene in Summit, New Jersey. I am currently an employee of AbbVie, Inc. The views here are my own. This is some background information on peripheral neuropathy, which is a common and debilitating adverse event often associated with chemotherapy. The sensory symptoms develop first in the most distal parts of the body, the hands and feet, manifesting as numbness, tingling, paresthesia, dysesthesia, painful sensations like shooting or electric shock-like pain, and in severe cases can lead to loss of sensory perception altogether. Sometimes this is also associated with motor symptoms like distal weakness, gait disturbance, fine motor impairment, and rarely with autonomic symptoms as well. Diagnosis of peripheral neuropathy can be made from a blood test to rule out a nutritional deficiency or underlying condition like diabetes or immune dysfunction, imaging to rule out abnormalities, structural abnormalities like a herniated disc or a tumor. Importantly, and what I'll get to later, are nerve function tests, electromyography, nerve conduction velocity, functional screens looking at reflexes, sweat tests, sensory tests, and the most invasive would be a nerve or a skin biopsy to look for structural or nerve abnormalities. Peripheral neuropathy is most commonly associated with anti-neoplastic drugs that act through various mechanisms. Shown here on the left are a few examples. In the mitochondria, oxidative stress associated with bortezomib, paclitaxel, and oxaliplatin can induce reactive oxygen species production, which can damage mitochondrial DNA. Cisplatin and oxaliplatin can also produce mitochondrial DNA adducts, and vincristine and oxaliplatin can also influence or affect mitochondrial calcium homeostasis. In the microtubules, vincristine and other vinca alkaloids bind the beta subunit of tubulin and inhibit microtubule formation, and paclitaxel and docetaxel, belonging to the family of taxanes, inhibit disassembly of tubulin, resulting in cell cycle arrest. And disrupted microtubules can affect retrograde and anterograde transport along the axon and affect membrane remodeling, resulting in length-dependent degeneration of axonal distal segments. Other mechanisms include neuroinflammation and myelin degeneration, which is often secondary to axonal degeneration, but can also be characteristic of Guillain-Barré syndrome, which is an immune disease affecting the myelin sheath of peripheral nerves. An underappreciated class of compounds that are associated with neuropathy are receptor tyrosine kinase inhibitors. Shown here on the right is a statistical analysis of clinical trials showing that patients taking VEGF receptor inhibitors are at increased risk of developing neuropathy in the clinic. And an analysis of 62 of the approved kinase inhibitors to date reveals that 30 of them are associated with neuropathies in the clinic. A case study is presented here for CC90003, which is a compound that was developed by Celgene, a covalent ERK1/2 inhibitor investigated as a therapy for solid tumors. So shown up top is the crystal structure of CC90003, as well as a graph showing that it is relatively selective, at least in this publication, for ERK and a few other off-target kinase families, which I'll get into later. This compound had efficacy in mouse patient-derived xenograft models, and specifically in KRAS mutant models. However, when this molecule went into the clinic, after 19 patients were treated in the phase 1A clinical trial, the clinical trial was halted and development of CC90003 was discontinued in part due to neurotoxic effects that were observed in the clinical trial, manifesting in a few different ways, Here is showing the incidence of dizziness, ataxia, which is gait disturbance, and dysesthesia that I mentioned previously is a telltale sign of peripheral neuropathy. Putting the clinical trial results in context, we look back at the non-clinical CC 90003 data. The pivotal studies used in the filing prior to the clinical trial included a 28-day GLP study in mice where no signs of neuropathy were observed, and a 28-day GLP study in dogs with a 28-day recovery, which had standard endpoints as well as nerve conduction velocity measurements, which were added due to clinical signs of neuropathy in the non-GLP exploratory study. In the GLP study, the same clinical signs showed up after 1 week of dosing with compound at the dose level of 16 mg per kg per day. And in addition to the clinical signs, the nerve conduction velocity of the sensory and the motor nerve at that dose level were significantly decreased during week 1 of dosing and week 3 of dosing. Notably, after dosing cessation, the animals did recover and the nerve conduction velocity returned to normal levels during study week 6. Further putting the non-clinical data into context, we compared the CC 90003 exposures that produced neuropathy in dogs and humans. We used 2 pharmacokinetic parameters, the maximum plasma concentration, or Cmax, as well as the area under the curve, or AUC, of exposure during the first 24 hours post-dose at these time points. And what we saw is that the exposures producing neuropathy in the dogs overlaps significantly with exposures producing neuropathy in the clinical trial by both Cmax and AUC, suggesting that 90003 elicited neuropathy at similar exposures in humans and dogs. Our research question asked whether CC 90003 induced neuropathy in dogs and humans was on target, dependent on RAS-RAF-MEK-ERK pathway inhibition, or was it off target? To ask this mechanistic question, we needed an in vitro model. Luckily for us, there are several published reports of models of peripheral neuropathy in vitro. This is just one example of an assay looking at neurite outgrowth. In this study, the authors derived human dorsal root ganglion-like neurons from pluripotent stem cells, cultured them for a period of time in the presence of a dose-response of chemicals, and compared effects on neurite outgrowth with effects on viability in the dorsal root ganglion neurite assay, as well as a similar assay using a central neuron from LUMIs. And they classified the compounds by effects on either the peripheral or the central or both neurons with similar potency. This is a very useful approach, but these cell types are not commercially available, which limited its applicability to us. There are a couple sources of commercially available peripheral neurons. This is one of them, published in 2017. This is the human-induced pluripotent stem cell-derived peripheral neuron from Ncardia called the peripheral cells. And using a very similar approach as on the previous slide, looking at neurite outgrowth, the authors were able to show sensitivity of a couple different classes of peripheral neurotoxicants, taxanes and microtubule interfering agents. However, they did not show sensitivity to platinum-containing compounds like cisplatin or a model protein kinase inhibitor, which led us to question the usefulness of the neurite outgrowth assay for identifying effects of kinase inhibitors. So our hypothesis was that a functional readout of neuronal activity may be more sensitive to a wider range of peripheral neurotoxicants. The assay that we developed internally used the peripheral U cells in the Axion Maestro system. So here we use the MEA plates, 48 well, with 16 independent electrodes per well. And here are just some immunofluorescence images from the published literature showing that these cells express MAP2, which is a neuronal marker, as well as peripherin, which is a specific marker for peripheral neurons. This is a workflow of our experimental design. We do coat the plates with PEI, which is standard for neurons. We plate on day 0, and we culture the cells for a total of 14 days on the MEA plates, taking readings throughout. And I should point out, an important aspect of our experimental design is that we start adding compound to our cells starting on day 2, and we replenish compound each time we change the medium. And so the cells are in contact with the compound for 12 out of the 14 days of culture. This is an important aspect of our experimental design because peripheral neuropathy often manifests weeks or months after the initiation of dosing. These are our initial validation results of the model with known peripheral neurotoxicants. We used 4: colchicine, which is a microtubule inhibitor; cisplatin, which is a platinum-containing compound; sunitinib, which is a receptor tyrosine kinase inhibitor; and thalidomide. We looked at 3 main metrics: mean firing rate over time in culture, the number of active electrodes in each well in culture over time, and the weighted mean firing rate, which is essentially the mean firing rate normalized to the number of active electrodes. We did see an effect of 3 out of the 4 compounds at a single concentration. However, with 2 of the compounds in particular, colchicine and cisplatin, we saw a significant decrease in cell viability over the 14-day culture, 12 days in contact with chemical. We did see that the effect of sunitinib on mean firing rate and weighted mean firing rate was a specific effect on electrophysiology with no corresponding cytotoxic effect. Importantly, the model system was sensitive to effects of CC90003. We did perform a dose-response characterization, and at this point I'd like to point out that throughout the 14-day culture, we see effects over time, and the easiest way to quantify those effects in time is to calculate an area under the curve of each metric over time at each concentration of chemical. So on the right, I'm showing the area under the curve for the weighted mean firing rate metric for CC90003 on the top, as well as another inhibitor of the same pathway, UO126, which is a MEK1/2 inhibitor, and both compounds potently inhibited weighted mean firing rate in this assay. The approach that we took to examine the ERK dependence of our in vitro findings was to evaluate the kinase selectivity of CC90003, sunitinib, as well as 12 structural analogs of CC90003. We screened many more than 12 in this kinase selectivity assay, but we selected 12 of them for downstream in vitro screening based on their varied selectivity of other off-target kinases. So you can see on the panel on the right, all of the compounds, with the exception of sunitinib, potently inhibited ERK 1 and 2 activity as well as BRAF activity. However, they exhibited a range of specificities against these other off-target kinases. We performed a dose-response characterization of all 14 compounds through the in vitro assay and compared our results to internal ERK IC50 biochemical data as well as the kinase selectivity data from the Z'-Light assay that I showed you on the previous slide. And what we found were 3 things. One, that the effect of the compounds on electric physiology was not correlated with ERK inhibitory activity, although we can't rule out ERK dependence just yet. It was not correlated with cytotoxicity either, suggesting that the effects of the compounds on neuronal electrophysiology were independent of cell cytotoxicity. But the effect was correlated with kinase selectivity using this metric of comparison called the Gini score, which is a metric used to compare selectivity of kinase inhibitors where the lower the score, the lower the selectivity, the higher the score, the higher the selectivity. And you can see now why we chose the 12 structural analogs of CC90003. They had lower and higher selectivity than CC90003, and notably sunitinib had the lowest selectivity of all the compounds that we tested. With our in vitro data in hand, as well as our Z'-Light kinase selectivity data, we had a tool set available to evaluate potential off-target effects on kinases besides ERK that may be correlated with in vitro activity of these compounds and may then also be associated with neuropathy. These are just a few of the correlations that we pulled out. The first example is FIR activity was significantly correlated with potency of inhibition of the number of active electrodes in vitro. IGF-1R inhibitory activity was correlated with the potency of inhibition of weighted mean firing rate. And PDGFRA activity was significantly correlated with viability IC50, suggesting that these kinases, inhibition of these kinases may be associated with these in vitro outcomes. We further interrogated the role of FIR kinase and IGF-1R on peripheral neurons using a literature search. FIR kinase is known to phosphorylate tubulin, is involved in microtubule dynamics, and regulates axonal retraction in dorsal root ganglia. Therefore, there is a plausible association between off-target FIR inhibition and the potency of inhibition of number of active electrodes that we saw in our in vitro assay, assuming that number of active electrodes is a metric of how far the neurites extend in vitro. There's significantly more information available on IGF-1R signaling in neurons. IGF-1R signaling is known to promote motility, neurite outgrowth, and survival of peripheral neurons, and IGF-1 inhibition decreases nerve conduction velocity in mice, which is a direct association with neuropathy. And furthermore, a single nucleotide polymorphism in IGF-1R was shown to be associated with early onset bortezomib-induced peripheral neuropathy, which suggests that if IGF-1R inhibition alone is not causative for neuropathy, it may be a risk factor for neuropathy caused by other mechanisms. I mentioned earlier in my talk that we couldn't rule out ERK dependence of CC90003 induced peripheral neuropathy. The reason is threefold. One, there are other RAS, RAF, MEK, ERK inhibitors in the clinic that are associated with peripheral neuropathy. Number two, all of our compounds in the in vitro assay did inhibit electrophysiological activity to some degree, albeit at varying potencies, which we believe to be a result of different selectivity against off-target kinases. And third, and this is a limitation of our approach, is that in the monoculture with peripheral neurons, we cannot capture the complexity of other cellular and molecular mechanisms of neuropathy involving other cell types like Schwann cells and endothelial cells. Schwann cells in particular are known to elicit ERK-dependent functionality, so it's plausible that 90003 induced a neuropathy by Schwann cell interference as well as neuronal electrophysiological effects. In the future, we'd like to evaluate a broader chemical set, including kinase inhibitors and chemotherapy drugs with known or lack of association with peripheral neuropathy in the clinic. But in conclusion, functional MEA-based measurements are sensitive to phenotypic effects of model peripheral neurotoxicants from this study, and future work will help bear that out. Like I mentioned in the beginning of my talk, this work was done at Bristol Myers Squibb, formerly Celgene, in Summit, New Jersey. This is a photo on the left of the group, non-clinical development. I'd like to acknowledge specifically my toxicology colleagues, Nate Collins, Steve Kopitek, and Kim Conley. While I was at Celgene, I worked in the investigative toxicology group headed by Kyle Colliget. A lot of the work presented here was done by our intern at the time, Caitlin Sudak, and I acknowledge the other members of the lab as well. Thank you for your attention. I look forward to the Q&A.
Thank you, Dr. Belair. That was terrific. Thank you to both of our speakers for sharing your work today. If you have any additional questions for Dr. Kiskinis or Dr. Belair about the research they presented today, please submit those now.